Sometimes it is very difficult to differentiate between the abiotic and the biotic stresses in the field because the symptoms produced by both kind of stresses are quite similar to each other. The situation is more difficult in case of plant viral diseases because the symptom produced by the plant viral diseases are quite related to the symptoms produced by nutrient deficiency. And second thing, we also kept in mind that unlike the fungi and the bacteria, viruses can't grow on the media. So by putting the infected tissue on the media will not confirm or reject whether the virus is present in the infected tissue or the not. To address this issue, plant virologists develop an easy, economical, but effective method which is known as the mechanical transmission of plant viruses. For this method, indicator plant is used, which is usually tobacco or the Chiropodium, which confirm the presence of virus or the absence of virus. This method is used for the multiple purposes. For example, to check the propagation of plant viruses or the infectivity of the plant viruses and also the host range of the plant viruses. And this method can also apply to test the resistance of any plant against the viruses. However, this method is dependent upon the condition of tissue from where the virus is going to be taken. Similarly, the condition of the host from where this infected tissue is taken, and also how the inoculum is prepared, and also the abrasive material. As abrasive material is used to create the wounds because we know that plant viruses themselves can't enter inside the host. So they need some kind of wounds, and these wounds are created through the abrasive material. For the mechanical transmission, you need infected plant which otherwise in a good condition, and also it must be a young plant so that you can get a good amount of virus from it when it is crushed. The second thing, you need two indicator plant. For example, here there are two indicator plant. One we called control, and the other we call the experimental plant. And these two plants must be same in every aspect. For example, genetically, age-wise, and must be grown under the similar conditions to get the authentic results. So these three are the prerequisite. An infected plant, leaf, or infected plant tissue, and the control as well as the experimental plant to perform the mechanical transmission. Plant viruses can be easily extracted by crushing the host tissue or the leaf with the help of pestle mortar. And when the leaf is crushed, the virus is released in the plant sap. But the issue is that not only the virus is released, but there is also the disruption of large and many other vacuoles, and their content is also added into the plant sap, which usually decrease the pH, so which is not favorable for the virus. So in order to stabilize it, it is always recommended to add the buffer, and the usually phosphate buffer is added to make the pH favorable for the virus. And not only the buffer is added, but also abrasive materials such as carborundum, 1% silica carbide, or the acid wash sand is also added in order to create the wounds. Sometimes carborundum powder is dusted before the inoculation, but there is an issue of inhalation of the carborundum. So it is recommended to add 1% silica carbide while you are crushing the plant material to get the virus. Once the extraction process is completed, then the plant sap is passed through the cheese or the muslin cloth in order to strain it. And the strained sample is collected in a separate beaker. And if the virus is the cause of the disease, then the virus must also be present in this infected sap. And if we put that infected sap on the leaf of the healthy indicator plant, it must produce the symptom. The inoculation process is very simple, both on the control plant as well as on the experimental plant. And it is just done by a gentle rubbing of the solution on the leaves of the healthy plant. The only difference in case of control plant and the experimental plant, that on the control plant you just rub a solution which contain the buffer and the abrasive material, while in case of the experimental plant you rub the solution which must contain the buffer, the abrasive material, as well as the actual infected sap, so that if the virus is present, it would show the symptom on the indicator plant. While in case of control plant, only the injury which is caused by the abrasive material can be observable. So these plants must be left under the control condition to observe the symptoms. Both set of plants left under the control condition to observe the symptoms. Initially, no symptom appear on the control plant, while in case of experimental plant, you can find the local lesion after 2 to 3 days of inoculation. However, necrotic spots appeared on the control plant because of the mechanical injury which is caused by the abrasive material, while in case of experimental plant, you can find the systemic symptom within 14 to 21 days. This is a very simple explanation of the mechanical transmission of plant viruses, and this process is usually adopted by the plant virologist all over the world in order to check the presence of virus in the infected tissue, as well as they also apply this method for many other purposes, such as to check the host range of the plant viruses, as well as to check the plant resistance against the plant viruses.